Greetings to Chapter 9. Welcome to Chapter 9, Controlling Microbial Growth in the Environment, Part 1. So this is just an overview of how we control microbes in the environment. So this is in your book, Table 9.1. It's called Terminology of Microbial Control. And I have some terms and definitions underlined. So for example, the far left column is the term column. Antisepsis is a term that you use to it's a term, but it's a process that you use to reduce the number of microbes on living tissue. So you use antiseptic techniques on living tissue. The next term, aseptic, refers to an environment that's free of pathogenic contaminants. So that means if you have an aseptic desk, then that desk is free of pathogens. And then these are suffixes, side and sidal. So for example, a bactericide is a chemical that would destroy a certain type of microbe. So a viricide would destroy a virus. This one's, I really like this one. This is so basic, degerming. It's really just degerming. It's mechanical, means when you wash your hands with soap and water, you are degerming your hands. The term disinfection, that means to destroy most microorganisms and viruses on non-living tissue. So you don't disinfect living tissue, you disinfect non-living tissue. Pasteurization, so everybody's heard of that, you get pasteurized milk. Sanitation, this is kind of a term that they use to meet public health standards. So I don't know if they do it on toilets anymore in hotel rooms, but they used to have a paper strip saying, sanitized for your protection. And then sterilization is where you destroy all microorganisms and viruses on an object. That includes destroying endospores. So these are chemical processes in this table. And then I just give definitions that we went over. So sterilization is removal of microbes from an object. Antisepsis can also be called germicidal. So that means the same thing. Destroys vegetative cells and viruses on living tissue. So you use germicides or an antiseptic cream, for example, on living tissue. Disinfection controls and destroys cells and viruses on objects. So you disinfect objects, you don't disinfect your skin. And then modifications of disinfection include degerming, sanitization, and pasteurization. So degerming, sanitization, and pasteurization are all types of disinfection. And then, for example, if you have an instrument that will come in contact with only the skin, so that'd be like a non-critical barrier of a patient, should be treated with a low-level antiseptic or germicide. This chemical process is continued. Pasteurization then, so we come down here, pasteurization, that's a process that kills non-spore forming pathogenic pathogens. So spore forming pathogenic pathogens will not be killed in pasteurization. And then we talked about sanitization. You just use sanitary techniques to lower the microbial load or the number of microbes in public health settings. So this is the terminology of microbial control and instrument classification. So here's the device classification. If you are a device used to control microbes, you could be a critical device, you could be a semi-critical device, or you could be a non-critical device. So a critical device would enter sterile tissue or the vascular system. All your organs are included as sterile tissue. So, and then your vascular system, that's just your blood vessels. So for example, a critical device could be a catheter used to, inserted in your urethra. Semi-critical device would be a colonoscope they'd use for a colonoscopy. A non-critical instrument classification, an example would be a sphygmomanometer. That's just a blood pressure cuff. All right, so let's talk about microbial death rates. So if you look at this graph, you have the number of living organisms, or excuse me, living microbes on the y-axis, and then you have the time on the x-axis in minutes. And then it shows you, so you start here at the top with 10 to the 9th microbes. So that's a billion microbes. And in each minute, this graph shows for every minute, 90% of the microbes that are on that surface you're trying to kill will die. So so let's see if we go here. So in this first minute here, you'll go from 10 to the 9th to 10 to the 8th. 90% of the microbes will die. So microbial death, the definition is the permanent loss of reproductive ability of microbes under ideal environmental conditions, and you have to define those. And you plot this on semi-log paper, so this is a log graph. Each one of these hash marks represents a 10-fold increase. So the microbial death rate is logarithmically constant under a particular set of conditions. Those are, for example, the ideal environmental conditions that you set as someone who's trying to disinfect or clean an object. And then it says here in the graph, a constant percentage of the extant, that's the living population, is killed each minute. So for example, you have a broth of microbes, start up here, 10 to the 9th, contains a billion microbes. You treat with an agent, under ideal circumstances, you treat with your agent of death, and in 1 minute, 90% of those microbes will be killed. So you go from 10 to the 9th to about 10 to the 8th microbes. And then you treat again for 1 minute. You'll go from 10 to the 8th to 10 to the 7th microbes left. 90% of the microbes will die each minute. So then you just treat again and again and again until all the microbes are dead. So let's look at the action of antimicrobial agents. So here's a cartoon of a bacteria. It's a bacillus, so it's a rod shape. It has its cell wall on the outside, then it has the cell membrane deep to the cell wall. It's got the chromosomes here in the middle, cytoplasm, so that's the intracellular environment. It has the ribosomes where it does protein synthesis. Here's a plasmid, so that's extra chromosomal DNA. And here's its little flagella that helps it move. And then this little guy down here in the bottom left is a virus. So here's the envelope on the outside, and then the green stuff is the capsid or the tegument, and then this yellow thing would be the nucleocapsid, and then the red stuff is the nucleic acid inside the virus. So one way to kill a microbe, you could alter the cell wall or the phospholipid bilayer, aka the plasma membrane. So you could damage the cell wall, so you could create an antimicrobial agent that would damage this cell wall. That means the microbe would be open to osmotic lysis, so it could lyse, it could burst. You could design an antimicrobial agent that damages the plasma membrane, the phospholipid bilayer, and then the cell contents would leak out, so the ribosomes, everything in the cytoplasm would leak out of the cell and it would die. If you have an enveloped virus, like in the cartoon down here, you could damage the protein in the envelope. So these little purple things are protein pieces in the viral envelope. You could also damage the envelope itself. so that the virus couldn't attach to the cell. You could, if you're designing an antimicrobial agent, you could also damage proteins and nucleic acids. So in this cartoon, here's a protein, it's a cartoon of a functional protein. If you damage the protein, and it's an important enzyme in microbial biochemistry, then you damage the protein. You could denature the protein, for example. The protein enzyme doesn't work now, and the bacteria can't do metabolism, and it dies. You could damage the DNA, so the bacteria then couldn't reproduce. So let's look at this, the factors affecting the efficacy or effectiveness of antimicrobial methods. Here's what you should think about when you're using an antimicrobial method to disinfect, for example, a surface of interest. So think about the site that's going to be treated. So you can't use harsh chemicals on living tissue. You have to use antiseptic techniques, germicidal techniques. So you have to keep thinking about, do you want to sterilize it? Do you want to disinfect it? You may have to sterilize items that are going to be used in surgery that will penetrate the outer defenses of the body. So for example, the skin, and go into your liver. Your liver is sterilized, so you have to use sterilized tools if you're going to penetrate the skin. If you're just going to be using the item to contact the skin or the mucous membrane, then you might just have to disinfect it. In the lab, we use 10% bleach to disinfect our surfaces. There's 3% chlorine in Clorox, and they sell it as 3% chlorine, and they use it as a household disinfectant. You may use that in your house. Okay, so this cartoon shows, so here's a bunch of bacilli. They're evil faces. If you don't use any antibiotics, the bacteria multiply. So here's a gang of bacteria. Bacteriostatic antibiotics prevent bacteria from multiplying. So the bacteria are still there, but they just can't multiply, so they can't make more. So if you've got 3 bacteria and you stop them from multiplying, they can't make 3 billion bacteria. That's bacteriostatic. If you are a bactericidal, then you actually kill the bacteria. See you, bacteria. So before you choose a physical method of control, you need to take into consideration all of these factors. You need to think about the time of exposure. How long are you going to be exposing this surface that you're trying to disinfect, for example? How long are you going to expose it to your disinfectant? Think about how much microbe is present on the surface. Sometimes you just have to guess at that. Think about the microbe that you're trying to kill. So MRSA, methicillin-resistant Staph aureus, it's resistant to methicillin. Think about whether the method you want to use is bactericidal or bacteriostatic. Sometimes bacteriostatic methods are good enough. You just stop the microbe from multiplying and eventually will die. Think about the relative susceptibility of microbes. So if you have, under a given set of antimicrobial methods and conditions, then the microbial death rate is constant. So the methods, how you're going to disinfect, is the surface a piece of wood? Is it tile? So under a given set of methods and conditions, you'll know, because scientists will have experimented under certain methods and conditions and found a microbial death rate, and you can find out what that is. But the microbial death rate will vary among different microorganisms. So for example, E. coli might not respond to the same antimicrobial methods and conditions as Staphylococcus aureus does. And then you have to take into account you may have a problem with resistant microbes. So remember, the Bacillus and Clostridium can form endospores. Mycobacterium has a waxy coating, so you have to take special consideration when you're trying to destroy Mycobacterium. Protozoa can be in a cyst form that will protect them from disinfectants. And then prions, which are infectious proteins, they're more resistant than any living thing. And sterilization does not inactivate prions, unfortunately. But they have come up with the new way to kill prions. I think it's called prionzyme. So I bet they're making a lot of money. Every mammal on Earth has a prion disease. In humans, it's called Creutzfeldt-Jacobs, and after a surgeon operates on a person with Creutzfeldt-Jacobs disease, they have to incinerate everything they use on that person because they have a prion disease. There's also spongiform encephalopathy in humans, and maybe you've heard of mad cow disease. That's a prion disease. If you've heard of scabies in sheep, that's a prion disease. So the most resistant, in this cartoon, the most resistant to being controlled by chemical methods, prions. They're really hard to control. But like I said, I think they have a new product called Prionzyme. The most susceptible to our methods are enveloped viruses. So selection of microbial methods continues. So in the environmental conditions that we were talking about, for a given antimicrobial method, the temperature and pH will affect the microbial death rate. So let's look at this graph. Here's the number of living microbes. So we start with 10 to the 5th. And here's the time on the x-axis. And then these are the temperatures. At 45 degrees C, it's the orange line. So you're using your antimicrobial chemical at 45 degrees C. You're using the exact same antimicrobial chemical, only it's at 20 degrees C. So for example, at 2.5 minutes, or sorry, what would that be? That'd be 3 minutes. Come up here, you only have this many microbes left. And at 20 degrees, you still have this many microbes left. So sometimes it's good to increase the temperature when you're trying to kill, or increase the temperature of your disinfectant. And then also acidic solutions are better because microbes don't like acid. So think about if you have to clean the surface before you try to disinfect it. So for example, feces or someone vomits, you want to wipe that off first before you put your disinfectant down. So how do you evaluate the disinfectants that you're interested in? We'll do this test in the lab, or if this is online, we'll do it online. I think the Kirby-Bauer test. But basically, it's a use dilution test, the Kirby-Bauer test is, and it measures the antibiotic effectiveness and minimum inhibitory concentration. So what you do in the Kirby-Bauer test, you have your plate here, and what you do, you dip these metal cylinders in microbes, and then you put the metal cylinders on the plate here, and then you dry, and then you dip the metal cylinders in different concentrations of a disinfectant, and you place them on the media, and then you see you take your antimicrobial or disinfectant of interest and you dip these in different concentrations of the disinfectant. And then you see which metal cylinder dipped in disinfectant, what concentration, kills the most microbes using the lowest concentration of disinfectant. So you don't want to, like from a monetary standpoint, if you're trying to sell this en masse, you want to use the minimum concentration of your disinfectant that will kill the most number of microbes. So you could use a really super high concentration of disinfectant, but it would cost a lot of money. But what if a lower concentration kills the exact same number of microbes? Then you want to use the lower concentration of the drug. Okay. And then you have these tests, other tests, Kelsey-Sykes capacity test. It's the standard test in the European Union. And what you do is you add, here's your disinfectant in this flask. You put some disinfectant in your sterile broth with the microbes growing in there. And then you take some of the microbes that have been mixed with disinfectant, and you grow them on plates and you incubate them. And then you see how many, you can see how well your disinfectant kills the microbes. And that's the end.